Hello. Today I'm going to be talking about point mutations. So point mutation occurs when one base is substituted for another base, and we have two types. The first one is a transition, where a purine is substituted for a purine. So an A can be substituted for a G and vice versa. Or a pyrimidine can be substituted for another pyrimidine. So C can be substituted for T and vice versa. The other one is a transversion, where a purine can be substituted for pyrimidine. So A can be substituted for T or C, and the same goes for G. Or a pyrimidine can be substituted for a purine, so C can be substituted for an A or G, and the same goes for T. So the effects that these mutations have on proteins. So here I have an example DNA sequence, and these are the amino acids that it codes for. So the first one we're going to talk about is a missense, and that occurs when a base is substituted for another base, and it changes the amino acid to a different amino acid. So when this G is changed to a C, tryptophan is changed to serine, and this would change the amino acid, which would change the whole DNA, the whole protein sequence. And this is an example of a transversion because a purine is changed to a pyrimidine. The next one is nonsense, when an amino acid is changed to a stop codon. So if this G were changed to an A, tryptophan would be changed to a stop codon, meaning our protein sequence would stop here and basically stop where it started. This is an example of a transition because a purine is substituted for another purine. Keeping up with the same example, the next one is silent. So that means no matter, like, when you change the DNA base, it still codes for the same amino acid. So when this T is changed to a C, it still codes for alanine and the protein sequence stays the same. And this is a transition because a pyrimidine this time is substituted for another pyrimidine. And the last one is a read-through, and that's when a stop codon is changed to an amino acid. So when this A is supposed to be green, this A is changed to a C, the stop is changed to cysteine. So instead of stopping right here, it would continue to go on, and we would have what would result in a longer protein sequence. And that is all.